we ready? They will let us.
Okay. So first of all, I really want to thank the organizers for giving me the opportunity, I see Maria Ren here, to talk here and to give a short presentation. I'm going to talk about recent work from my lab. So we are working, so that the title is Coordinating Chromosome Segregation with Cell Cycle Progression in Oocyte Meiosis. And what you have, so it's really nice to talk actually at a meeting like this where everybody has introduced meiotic maturation already. So we are really interested in the first and second meiotic divisions. So these are the last steps of gametogenesis, and they take, as you all know, place in the adult female. So the overarching questions of what we are interested in my team are to understand how meiosis I events are implemented only in meiosis I, and meiosis II events only in meiosis II, meaning there are, for example, things like in meiosis I you have to suppress S phase when you exit meiosis I. However, after meiosis II you have to allow exit from the meiotic cell cycle and S phase. On the other hand, fertilization arrest has to take place only in meiosis II but not meiosis I, and all these things are super important to generate oocytes that can be fertilized and will give rise to healthy offspring. Now today, I'm going to talk about stepwise cohesion removal. So as you all know, in meiosis I, it's chromosomes that are segregated, and in meiosis II, it's sister chromatids. And in both cases, the chromosomes and the sister chromatids are held together by cohesion. So cohesion on arms is holding the chromosomes together because on arms they have recombined. And in the centromere region, it's only meiosis II that sister chromatids now are held together. So it's important that cohesion is maintained up to metaphase because this is really what allows to the establishment of tension-bearing attachments. And what is really important also to keep in mind is that in both meiosis I and in meiosis II, it's the same enzyme that's removing cohesion from arms in meiosis I and from the centromere region in meiosis II, which is separase. So the stepwise removal of cohesion requires thus that separase is activated twice, once at the metaphase to anaphase transition of meiosis I, and the second time at the metaphase and anaphase transition of meiosis II. And to prevent precocious cohesion removal, it's really important that separase control is very tight and doesn't occur, for example, at the transition from meiosis I into meiosis II or before metaphase to anaphase transition. Now, we have also so what is really, what allows this stepwise cohesion removal, and what is really important is also that so cohesion at the pericentromere, which I will call centromeric cohesion, is protected from cleavage by separase throughout the first meiotic division. So this protective mechanism here is what we call pericentromere cohesion protection. So this is put in place at a certain time and is removed at a certain time. But what is really, really important, it's in place when separase is active in the first meiotic division. Now, why we are interested in so what we really want to understand is how separase control and pericentromere cohesion removal and pericentromere cohesion protection are coordinated in oocytes. And to better understand this, we would like to understand, get conditions where we have separase active at a time when separase is usually not active, to see whether cohesion here is protected, and also to see, to really see when this pericentromere cohesion cleavage, pericentromere cohesion protection is in place. And I just have to go back. So why we are really interested in this is, so as you have heard already by Eva's talk and also the following talks, is that missegregations in human oocytes are really error-prone, and most trisomies are actually due to missegregation errors in female meiosis, and most of them take place in the first meiotic division, but also in the second. And we have also this staggering curve here, which increases. So you have seen that from Eva's talk, and this is also taken from a review from her lab. You see that this increase with maternal age of missegregation events, and what is really important to point out, and what she pointed out as well, is that it's actually certain missegregation events that increase with age, which are precocious sister chromatid segregation and what she called reverse segregations. In both cases, this is due to loss of pericentromere cohesion, meaning that you start to segregate sister chromatids. So there is cohesion weakening with age that has been shown in mice and in humans, and this extensive cohesion weakening and this loss of centromere cohesion with maternal age really correlates with this high error rate with age in human oocytes. So as I said, now to understand separase has to be inactive before metaphase, has to be tightly controlled before metaphase to anaphase transition and between the two meiotic divisions. And at the same time, cohesion protection has to be put into place. So this is happening before metaphase to anaphase transition, but we don't know when exactly. And then it has to be removed at a certain moment before metaphase to anaphase to transition upon fertilization. To obtain constitutively active separase, to be really able to see when cohesion is protected, we focused on so there are two main separase inhibitors, which are saccharin and cyclin B1 CDK1. So we use different tools to remove those two main inhibitors of separase in meiosis, in oocytes. So on the one hand, we have a saccharin knockout. This is a complete knockout, so mice are fertile and have no overt problems. And we use a separase knockout, a conditional knockout, which we replenish with a separase mutant that cannot be inhibited by cyclin B anymore. So we can combine those two mouse strains, these two approaches. So we have a saccharin separase double knockout oocyte, where we introduce the cyclin B1 CDK1 deficient separase mutant, inhibition deficient mutant, where we have separase which we call out of control. So what is happening? We also need a tool to see actually what's happening, so whether separase becomes activated. So we have a tool where at the one time we can measure separase activity and also follow chromosome movements. So this is a biosensor, which is based on a biosensor that has been described in mitosis by the Hirota lab, and which we adapted to mouse oocytes. So in principle, we have a separase substrate here, which is sandwiched between two fluorochromes. And upon activation of separase, it gets cleaved. We lose thus the co-localization. The whole stuff is localized to the chromosomes, so we can follow the chromosome movements and we can quantify the signal. So we can do these movies. So this is here in meiosis 1. And you can see this change of color once anaphase takes place. So this is really very clear to follow chromosomes and follow separase activity at the same time. Now in meiosis 1, what is happening if we put active separase here? So we have this condition here, and now we have separase that in the double knockout oocytes that is active. So if we have just the double knockout oocytes, nothing is happening because we don't have any separase, so there's no change of color and chromosomes remain aligned. Now if we introduce wild-type separase, what you can see here, so chromosomes move around, they align, and then you have a separation with the change of color. Now, if we introduce the separase mutant, so now we have separase that's not inhibited either by cyclin B1 nor by saccharin. What you see that things go really bad really quickly. So immediately after resumption of meiosis, we see this change of color, and chromosomes are all over the place. We don't even see a correct anaphase. So, and it looks like much more than just chromosomes that have been separated, which brought us to the question of whether pericentromere cohesion hasn't been protected at that moment. And indeed, when we did chromosome spread, so here you have the control conditions with no rescue, so we have only bivalent, so paired chromosomes present in these typical cross-shaped figures. In the rescue conditions with the separase mutant, you just see mainly single sister chromatids, meaning that pericentromere cohesion has been cleaved, so it has not been protected under these conditions. Active separase here, if you have constitutively active separase, this is enough to separate, to cleave all cohesion. So what we can say from this result is that at resumption of meiosis, all separase cleaves all cohesion and we get single sisters, whereas if we activate separase later on, we just cleave arm cohesion, whereas centromere cohesion remains protected. So meaning that cohesion protection is set up with time, and we could actually confirm this by looking for the proteins that protect separase, and we were able to show that they are recruited only later, only in prometaphase to the centromere region. So this means really there is no protection. This is a weak point actually. So if there is leaky separase activity, it might lead to weakening also of pericentromere cohesion at resumption of meiosis. Now what happens now in meiosis 2? So in meiosis 2, this is a really very special cell cycle state because oocytes are arrested, but they have to be able to immediately activate once the sperm arrives. So it's a very transient kind of thing. So the inhibition has to be very tight, but immediately we have to be able to reverse it very easily as well once fertilization occurs. So we took metaphase 2 oocytes. We injected again the separase mutant. So here this is for simplicity. I show the data with the saccharin knockout, and what you can see again. So this is without activation. We immediately see sister chromatids coming up. And we can do movies of that. So this is because there's a lot of autofluorescence. I show it only for the saccharin knockout. So it's only the DNA that you can see here, not the sensor. But you see that chromosomes just fall apart. So there are no real anaphase movements, and just the sister chromatids fall apart. So now this was really indicating somehow that this occurs without any anaphase onset, which actually was also unexpected because it was thought that actually pericentromer cohesion is still protected in the metaphase 2 arrest and would require cell cycle progression to actually be removed. Now, cell cycle progression, this means activation of the APC or the anaphase promoting complex. So we were wondering whether under these conditions the anaphase promoting complex was getting activated for some reasons. And so as a readout, we have a separate APC substrate that gets ubiquitinated and degraded, so we lose the fluorescence, and this is what you see in the control after strontium activation. However, when we inject this mutant and we look out for the APC substrate, you can see it remains completely stable, meaning that there hasn't been any cell cycle release. So what we see here is centromeric cohesion cleavage without anaphase onset, also in meiosis 2. So that means, in contrary to what was expected, what would have been something like where you think it would make sense for an oocyte to be able to protect precautious cleavage of pericentromere cohesion, what we see here is actually there is no protection in place. If we put in constitutively active separase here, in metaphase 2, we just get separation. We get separation of sister chromatids in a completely unordered manner, same as when we activate oocytes for metaphase to anaphase transition. So there is no robust centromere cohesion protection present during this metaphase 2 arrest. Now, just to finish up, we have found that cohesion protection here in blue is in place at the metaphase to anaphase transition in meiosis 1, but not in early meiosis 1 at resumption of meiosis, and not during this meiosis 1 to meiosis 2 transition, so these are data I haven't shown, and also not during this metaphase 2 arrest, so it's removed actually quite early, immediately after exit from meiosis 1. And data I haven't shown are that we found that the main inhibitors of separase in meiosis 1 up to metaphase to anaphase transition are saccharin and cyclin B1, as well as in meiosis 2 up to metaphase to anaphase transition. However, between meiosis 1 and meiosis 2, it seems that the main inhibitor of separase is cyclin B1 and not saccharin. And with this, so if you're interested to learn more about it, this paper has been deposited on bioarchives. And I just want to thank the people involved in this work. So this has been the work mainly of an outstanding PhD student, Safia El-Chayani, who is going to finish and will be on the job market. And with contribution of these members from my team, Elvira, who's now in the lab of Marie-Hélène Berlac, who has contributed in the very beginning. And last but not least, I want to finish by saying that we are looking for postdocs. So if you're interested in this project or on CSF arrest, please come and see me, or if you know anybody, please let me know.
Okay, we have time for one question from the audience.
No, what we think actually what is happening, so and this we have published, is that when you're in anaphase 1, there's a separase substrate, which we don't know yet what its identity is. It could be either mycin or a special fraction of Reg8, which is cleaved by separase and which leads to a physical separation of the kinetochores, the sister kinetochores, and we found that that is required to bring about deprotection. So we think that this is the key event. However, for right now, we don't know what the substrate really is.
Okay. In the interest of time, we should move on. Let's thank Katja again for her talk. And our next speaker is Thierry Lorca from the Center for Cellular Biology in Montpellier.
Okay, first of all, I would like to thank the organizer to give me the opportunity to speak today. So in our lab, we are interested on the mechanisms that control mitotic entry and its progression. And this even are under the control of the silica-1 cyclin B kinase activation that is negatively regulated by the WEE1-MYT1 kinase that phosphorylates silica-1 of the tyrosine 15. When cells are satisfied, the phosphatase CD25 is activated, dephosphorylates this site, and promote the silica-1 cyclin B activation that promote a lot of phosphorylation required for this event. But we know that beside silica-1 cyclin B activation, we need to inactivate the phosphatase PP2AB55 that counteract the phosphorylation on silica-1 cyclin B. Okay. Okay, thanks.
Sorry.
So this phosphatase counteracts the phosphorylation of silica-1 cyclin B on mitotic substrate. And this pathway that inhibits PP2AB55 is driven by the grade-wall kinase that phosphorylate two substrates called NSAR and HARP19 that one phosphorylated bind and inhibit PP2AB55. We and others have been involved in the work about the role of PP2AB55 in grid-wall during mitosis, but its implication during meiosis is less well understood. And today my talk is about this topic. So to do that, we use the Xenopus oocyte, and these oocytes are first arrested in prophase 1 or meiosis for years. And the progesterone releases this arrest. And 3 and 5 hours later, the oocyte reach GVBD and metaphase 1. And in Xenopus, it's easy to score oocyte in GVBD because we can see a white spot in the dark animal pole. Next, cyclin B are degraded between meiosis 1 and meiosis 2, and translation of cyclin B reach meiosis 2, where the protein ERP1, an inhibitor of APC, bind APC and maintain the oocyte arrested in meiosis 2, in metaphase 2, until fertilization. So to work about the grid wall and the role of grid wall in meiosis, we use a TRIM21 protein. We microinject the mRNA coding with TRIM21 and the antibody that detect or bind the grid wall, and TRIM21 binds the antibody, ubiquitinate the antibody and grid wall, and the complex is degraded by the proteasome. Grid wall is expressed in prophase 1 of meiosis, phosphorylated after progesterone addition in meiosis 1. This phosphorylation is correlated with its activity and associated with the phosphorylation of PLX1 that promotes the activation of C25 that dephosphorylate silica-1. When this site is dephosphorylated, it means that silica-1 cyclin B has been activated. The oocyte, Xenopus oocyte in prophase 1, and in meiosis 1, you can see the white spot that is easy to detect and to score. When we deplete the grid wall with the TRIM21, it's an efficient degradation. And when we score meiotic maturation and we compare the control and the oocyte devoid of grid wall, we didn't find any differences. This result is also in Western blot. We can see that PLX1 is phosphorylated in control oocyte and in oocyte devoid of grid wall. Silica-1 has been activated. As a readout of silica-1, we can follow this phosphorylation of PP1 that is phosphorylated. So it was difficult to believe that without grid wall, the oocyte can resume meiosis without mistake, and performing a phosphoproteomic analysis, we find it here in this 10 replicate in Xenopus devoid of grid wall that we have a lot of dephosphorylation. each band corresponds to one site. Among this dephosphorylation, we detect two sites that belong to the MAP kinase protein in the threonine and tyrosine. We can see here the dephosphorylation in GVBD and metaphase 2. And this MAP kinase plays a central role during meiosis. This protein is phosphorylated indirectly by MOS. MOS is a protein translated after progesterone. MOS activate MEC, that activate MAP kinase. MAP kinase phosphorylate RISC, that phosphorylate and stabilize ERP1 to inhibit APC and to maintain the oocyte arrested in metaphase 2. We confirm that this phosphorylation does not occur anymore when grade wall is degraded. This dephosphorylation is specific of grade wall because when we add back grade wall, we can see that we can maintain the phosphorylation in this condition when we rescue the protein. To make a long story short, we have shown that this dephosphorylation of MAP kinase is due to the instability of the non-translation of MOS. MOS is not anymore translated in the absence of grade wall, and you can see also that ERP1 that is stabilized in control condition is not anymore present here. When we rescue MOS, MAP kinase, and RISC in this condition. Next, we try to see whether by overexpressing MOS we can recapitulate this pathway. We can see that overexpression of MOS can indeed activate MAP kinase, can also activate RISC protein, but it failed to stabilize ERP1. You can see ERP1 that is stabilized in control condition, destabilized when grade wall is depleted, but MOS is not able to stabilize ERP1. So this result means that the grade wall control an additional mechanism able to stabilize ERP1. And recently, it has been shown that silica-1 cyclin B3 by phosphorylating ERP1 can also target ERP1 for degradation. And the next question was to see whether grade wall can modulate the amount of cyclin B3 during meiotic maturation. And as expected, in control conditions, cyclin B3 is degraded. But when we deplete grade wall, this degradation does not occur anymore. And again, grade wall addition add back this degradation. So unlike MOS that was enabled to stabilize ERP1, we try to see whether depletion of cyclin B3 can maintain ERP1 in the absence of grade wall. And to see this experiment, we perform a double degradation with TRIN21, the grade wall and cyclin B3, and you can see that even in the absence of MOS and MAP kinase, ERP1 is stabilized again in the absence of grade wall. And this stabilization is enhanced when we add back MOS, meaning that both pathways are involved in this condition to maintain ERP1 stabilized. So to see and to understand why cyclin B3 is not degraded in the absence of grade wall, we have done some work, but we also knew that cyclin B3 was degraded by APC dependent manner, and we see whether grade wall can regulate also the APC. And you can see here, in control condition, in the absence of grade wall, we follow the phosphorylation of APC3, one component of the APC, that is phosphorylated in meiosis 1 and meiosis 2, that is not anymore phosphorylated in the absence of grade wall. Associated with this unphosphorylated form, we also maintain the cyclin B2 in a non-degradation form compared to the control condition. We can see also the degradation of cyclin B3 that is stable and the fact that we can maintain ERP1 in its stabilized form. Associated with the stable phosphorylation stability of cyclin B, you can see that silica-1 cyclin B activity is higher in the condition where we deplete the grade wall compared to the control condition where we have fluctuation of silica-1 cyclin B between meiosis 1 and meiosis 2. And to finish, and we check also what we obtain in phosphoproteomic analysis, we see that APC1, another component of APC, that it's important for the APC activity, these 3 sites are dephosphorylated in the absence of grade wall. We confirm in Western blot that these sites are dephosphorylated in oocyte devoid of grade wall. You can see in control conditions the phosphorylation site, dephosphorylated when grade wall is not anymore present in the oocyte, and phosphorylated again when we add back grade wall. So we conclude that grade wall, by inhibiting PP2AB55, provides the correct temporal activation of 2 pathways, APCC and CMOS accumulation, 2 events essential to restrict cyclin B3 dependent degradation of ERP1 before JVBD, and to allow stabilization by the MOS MAP kinase pathway. I would like to thank Thierry Lorca that he was involved in this work, Céline Benchung, Benjamin Lacroix, Anna Castro, some collaborators, and also some people who are working on MRE in the lab. Thank you for your attention.
Thank you, Thierry. We have time for a couple of questions.
Thanks, Thierry. So actually, I'm a bit lost. So they are arrested in metaphase 2, these oocytes, or?
In the absence of grade wall? No.
Okay.
No. Since when you deplete grade wall, you cannot degrade cyclin B3. So degradation of cyclin B3, it's a readout of the meiosis 1, meiosis 2 transition. So maintaining cyclin B3, you maintain the oocyte in meiosis 1. So now, really, we don't know if we have spindle or what is the phenotype in the spindle without grade wall. What we know also that the polar body is not
extruded. Okay.
Sorry. I was wondering, so maybe I missed it because it was really fast. So you think that grade wall would work on the MOS-MAP kinase pathway and that would feed back on cyclin B3, right?
Yes.
But what is your proof that it's not the other way around, that grade wall would play on cyclin B3, that would play on the APC, and that would feed back on the MOS-MAP kinase pathway?
Because when we deplete APC, just APC, and next when we deplete cyclin B3, in this case we maintain ERP1, okay, and MOS and MAP kinase are activated in this condition.
All right, let's move on. Let's thank Thierry again. And our next speaker is Tommaso Cavazza from University of Zurich, and who would tell us more about genome unification is error-prone in mammalian embryos.
I guess the high is good. Okay, thank you for giving me the chance to present my work. And I slightly changed the title as I decided to present you also something new we're working on the lab, and it's actually a new concept. I will be very happy to have feedbacks on this. Yeah, so my lab we work on embryogenesis. You are all aware that embryogenesis starts when the sperm fertilizes the egg. But what I want to point out that is in the zygote, the 2 parental genomes are actually kept separated into individual nuclei called pronuclei, and only at the onset of mitosis, at nuclear embryo breakdown, they will actually merge for the first time. Then you have multiple cell divisions, morphogenetic events, cell differentiation, and you may organize a blastocyst. This is, however, not really efficient in humans. We already heard it. It's about 50% of an embryonic failure. I also find it very fascinating because it's strikingly clear. Anyone, even not a trained person, can see which one is the good one and which one is the bad one. I actually use this to test the students when I interview them now. And I was really interested in this type of topic, and so I was a postdoc in Melina's lab and in Germany, but also in Switzerland, where we are now. You cannot work with human embryos, but even in countries where this is possible, it's, the amount of samples available is really limited. And so when I was in Melina's lab, we established a method to image bovine embryos using live microscopy. Making the embryos, it was actually something made more or less in a routine way by the veterinarian departments. But we implemented the live microscopy. This is how it looks like. In this zygote, you see the two pronuclei here. It's DNA labeled by histone H2B, and there will be also the microtubules labeled in white here. And the system works quite nice, I would say. And there were many observations one can do on this kind of videos, but what really caught our attention and then we focused on, it was what happens at the onset of mitosis. Actually, in the hour before mitosis, if you look at these stills from the video you have just seen, the two nuclei are not really fully used by the chromosomes. They are clustering at the interface between the two pronuclei. And we call this process parental genome clustering. And if you're a mitosis aficionado, you may also notice that the chromosomes are really highly condensed at the onset of mitosis, which is also interesting. And we, however, also checked whether this process was efficient, and what we found is that in about 20% of the cases, one of the two pronuclei was yes polarized, but outward, as in this example, and another 20% of the cases, there was one nucleus that we term uncondensed, where the chromosomes were not so condensed at the onset of mitosis, and they were more spread out inside the cytoplasm. And then we, next we thought about the frequency of mitotic errors. So I quantify chromosome congression defects and chromosome segregation defects, and see whether these three groups have different amounts. And indeed, the cluster group presented only 20% of mitotic errors. I still think it's actually pretty high. The unclustered group was 50-50, and the uncondensed group, it was all displaying really massive type of errors. You will see this later. And we wonder what could be the causes for this, and we thought to a geometrical hypothesis. And as these two nuclei are huge in comparison to somatic cell, by clustering the chromosome at the interface between the two pronuclei, this may reduce the space used by the chromosome, maybe speed up spindle assembly. And then this would mean that chromosomes that are located here or in this nucleus will be actually missegregated. And I tested this in multiple ways. I will show you just one data. So this is the unclustered group, and you should follow the chromosomes that are in this yellow circle and see that they will actually join the spindle later. And one of them, for whatever reason, very happy to figure out why, fails to really join the metaphase plate, and it ends up in a micronucleus. And we further work on this, and to save time, I just joined to the conclusion, which is that the process of parental genome clustering requires three key steps. The first one is to polarize the pronuclei right after pronuclear formation, and this yellow or orange trace here, if you can see it, this represents nuclear pore complexes which get polarized, and then the two pronuclei migrate together thanks to the action of the microtubules, and then they have start to orient to kiss each other. And what I find interesting in the context of this symposium is that parental genome unification seems really to be triggered by the activity of one component of paternal inheritance, the centrosome, and the second one is of maternal inheritance, which is the nuclear pore complex, which is really bridging the cytoskeleton, microtubule cytoskeleton to the chromosomes. However, I should also remark that this process is really inefficient, and this is why we think mitosis is often error-prone in bovine and human zygotes. And this work was done in the lab of Melina, also thanks to a lot of collaborators that helped the work, and I would like to acknowledge them all. And now I would like to take the second half of my 10 minutes to tell you about what we are doing in the lab. I started around 2 years ago, and we really want to push forward in the bovine model, but also to focus on what is causing embryonic failure. And we're actually now trying to see it from a different angle, and we're reasoning on whether embryonic failure is actually an active mechanism that is triggered by a sort of quality control mechanism. Why would you need a quality control mechanism? Well, both, at least human and bovine, both species have around 9 months of pregnancies, and it's usually a single embryo, and thus we have a sort of pressure to prevent pregnancies of unhealthy embryos. And what is this 50% of embryonic failure is actually an active process that when an embryo is defective is pushed towards failure. And one can think of another way to reformulate this: what happens if one of the two gametes is contributing a damaged DNA, and how does the embryo deal with it? So together with Ying, which is a PhD student in the lab that was brave enough to join me, we went back to look at this uncondensed phenotype. As I mentioned, and as you can see in this video, there's really a massive number of chromosome segregation problems. You see, okay, the video is a bit not right, but you would see a lot of lagging chromosomes between the two spindle walls. And what we thought is that, it could actually be that this nucleus is actually carrying DNA damage, and then the activation of the of the DNA damage checkpoint is blocking progression selectively in this nucleus. So what we did with Ying was initially to clone a reporter for a live reporter for DNA damage, for the DNA damage response, and it turned out that RAD51 in our hands worked pretty well. And here you can see that in this specific nucleus here, we detect quite a number of RAD51 foci. They decrease over time, but shortly before the onset of mitosis, they're still present, indicating still active DNA damage response. And the other nucleus, on the other hand, is free of foci, and it will enter mitosis. And so we have, again, this asynchronous chromosome condensation that in most of the cases also results in a synchronous NEBD. But now with this reporter, we can quantify the number of foci, and as you can see here, in the delayed nucleus, actually, we frequently see an elevated number of RAD51 foci. So we decided to push it a bit further, and what we did, it's probably a very brutal experiment. We take either the sperm or the eggs, put them in an X-ray machine, irradiate them with 10 gray, it's really a lot, and it's crazy that the germ cells still retain the ability to fertilize or being fertilized. And, but then the phenotype was very clear. So if you have this highly damaged sperm in this specific image, you see a lot of these RAD51 foci, but then most of them undergo this asynchronous chromosome condensation. And then we went back to the cell cycle literature, and the cell cycle literature says that each nucleus can go through the cell cycle independently inside the cytoplasm. But in theory, when the mitosis, one nucleus enters mitosis, this is a signal that will override everything else. So the rise in CDK1 activity will push every nucleus into mitosis. And so if this is the case, then this also the delayed nucleus will be pushed into mitosis. And so what we thought is that by inhibiting the DNA damage checkpoint, we will let this nucleus to progress through the cell cycle and eventually enter mitosis with the same time. And so what Ying did was to take these cells, incubate them so take zygote generated after irradiation, incubate them in CHEK1, CHEK2 inhibitor, and indeed most of these embryos will enter mitosis with synchronous chromosome condensation. This doesn't mean they are healthy, they're carrying a crazy amount of DNA damage, but still we completely rescue this phenotype. And so what we think is happening is that indeed when parental genome is damaged, it activates the DNA damage response. This blocks the cell cycle progression at a local level. So we get this kind of configuration where one nucleus is progressing, is about to enter mitosis, but then there's a synchronous mitotic entry with chromosomes that were not prepared, actually leads to massive chaotic chromosome segregation, and this will clearly lead to embryonic failure. And I'm actually closing with a question, and I'm proposing that this is a mechanism of quality control, because it's really a system that it's made to fail. So the embryo prefers not to repair the DNA, but actually to undergo these chaotic divisions and embryonic failure. So with this, I'm done. I would like to acknowledge all my team, especially Ying, and our collaborators, funding agency, and you for your attention.
We have time for a couple of questions.
Hi, thank you for the talk. I was wondering if the pronuclei that shows this uncondensed chromatin was always like coming from the dad or from the mom, or if it was like a 50-50.
So this is interesting. So in Melina's lab, I had 70-30, so 70% male, 30% female, and now we have actually more frequently the male. So it's almost 90% male. There are two possibilities, probably both contributing. One is the sperm is different, so we are working with another bull. And the second one is also the oocytes. I think we have a slaughterhouse that is really close to our lab, so maybe they're a bit less stressed. And so I think it's also very interesting in terms of IVF procedures, how much germ cell stress can actually contribute to impairing the ability, some abilities of the than embryo to repair DNA, for instance. Yeah.
Gabri?
I can shout.
No, I think they don't want it for the stream.
Thanks. Excellent talk, Tom. Quick question, what happens, so you showed irradiated sperm DNA, right? What happens to when you irradiate the oocyte DNA?
It's specular. So then you will
so
have...
it also has the effect?
The endogenous problem is mostly in the male. It's well documented. Sperm frequently has DNA fragmentation. But so if we irradiate with 10 gray the oocytes, then you will have this massive amount of DNA damage in the oocyte, and then still sometimes in few cases they do enter mitosis synchronously because also the male is damaged, but usually then it's always the female being delayed. So it seems to be like a similar cycle response.
Okay. Thanks.
We have a question here from Marzia.
Thank you. Do you know if there's a threshold effect, like a certain level of DNA damage that will trigger the quality control?
We are actually trying to think on it, but it's very difficult to address it. I mean, you can do it experimentally by increasing amount of gamma, gray radiation. We indeed, when we use 5 gray, it's less penetrant, and so that's why we went so high. But it would be nice to know how many breaks there are, right? And it's kind of very difficult to quantify at a single cell level, at least to my knowledge. And so, yeah.
So in your design to fail model, so is the egg in control here, or is the male also contributing to the failure? Is the egg really picking the sperm that it wants, so to speak, according to your model?
I would say that, I mean, I should think on it a bit more in depth, I think. But I think it's overall, it's a maternal contribution in a way. I mean, I think in this case, it really looks like that the sperm is contributing potentially a damaged DNA, and then the cell cycle machinery that is basically provided by the egg, right? It's reacting in this way. So yeah, I would say that is how the egg cytoplasm deals with the intruder. Yeah.
A nice talk. I was wondering if this decondensed phenotype is transient, or will it stay decondensed even if you delay mitotic entry?
I really want to do this experiment. So yeah, I think it's a bit tricky to to really time at the right time to add the Rho inhibitor, but I think it's what we have to do. So basically, I think the question is, if you give enough time, will eventually the damage be repaired or not? And yeah, it's a very good question. Yes.
All right, let's thank Tom again then.
Thanks.
And our next speaker is Christopher Thomas from the Institute for Developmental Biology in Marseille, who will reveal us the secrets of ovulation.
Good afternoon, everybody. Firstly, thank you very much to Elvin and Marie-Hélène for arranging such a wonderful conference. I'm Christopher Thomas, and today I'll talk to you about our work into revealing the secrets of ovulation. Here we can see an egg. All new mammalian life originates when an egg like this one is fertilized by a sperm. But before this fertilization can take place, the egg must leave the ovary from the ovarian follicle in which it is developed. This process is known as ovulation, and without it, no new life is possible. Here we can see this ovarian follicle positioned on the edge of the ovary. It has the oocyte, or immature egg cell, at its center, surrounded by many specialized cells whose job it is to both help the oocyte develop, as well as to ensure its safe passage out of the ovary during ovulation. Once during each ovarian cycle, we have a systemic increase in luteinizing hormone, which causes a dramatic remodeling of the ovarian follicle, the end product of which is that the follicle wall ruptures and the egg is released to the outside. And while we understand that ovulation is initiated by luteinizing hormone, the exact pathways that drive it forward and its regulation in space and time have remained unclear, because ovulation takes place deep inside the body, making it incredibly difficult to study directly. In this work, we wanted to ask, what is the mechanism of mammalian ovulation? And to allow us to do this, we developed a model system to visualize ovulation live in single isolated antral follicles from mice. Here we can see one of these antral follicles. It has the oocyte at its center, and we can see DNA labeled in magenta, cell membranes in green, and as well as this with micro-injected fluorescent dextrin, shown here in orange, to mark the fluid components of the follicle. On addition of HCG to stimulate ovulation on the microscope, we observed a number of discrete phases that take place synchronously across all follicles. Those are that first the follicle expands, the follicle then contracts, ruptures, and the egg is released to the outside. And this is taking about 12.5 hours in time with when ovulation takes place in vivo in this mouse strain. So once we had ovulation working on the microscope, our first question was, does this accurately reflect in vivo ovulation? And to answer this, we used comparative single-cell RNA sequencing, where we took follicles from our ex vivo system and compared them against follicles isolated from stimulated mice at time-matched points. And to go through a lot of data very fast here, we focused on a number of key ovulation-related genes and showed that their temporal expression was the same between the two data sets, and furthermore, global gene expression was highly overlapping throughout ovulation. So we're happy that our system is accurately reflecting in vivo ovulation. We then wanted to describe ovulation in a quantitative manner. And to do so, we used two complementary imaging strategies. The first was confocal microscopy, in which we opted for a maximal resolution in X and Y, but just imaged 1 or 2 slices through the height of the follicle. And this allowed us to see this detailed cellular dynamics deep inside the follicle. The second strategy was to use 2-photon microscopy with a much lower resolution in X and Y, but instead imaging this detailed Z-stack throughout the entire height of the follicle, allowing us to generate 3D surface reconstructions and describe transitions in both follicle size and follicle shape through ovulation. And one of the key measurements we made was to look at follicle volume. So just like in the video we saw before, we observed that for the first 8 hours, we see the follicle increase in volume as it undergoes expansion. Between 8 and 12 hours, it decreases in volume as it undergoes contraction. And finally, at 12.6 hours, it ruptures and the egg is released. So now I'll focus a little bit on some of the work we did to identify the pathways that are driving each of these phases of ovulation, starting with expansion in the first 8 hours. So we first asked ourselves, what do we know is happening in the follicle at the same time that we see follicle expansion? And we know that the cells that directly surround the oocyte, the cumulus cells, spread outwards in a process known as cumulus expansion that's driven by hyaluronic acid secretion. We hypothesized that this might be important in follicle expansion and wanted to quantify its dynamics in the context of the whole follicle. So to look at this, we used cryosectioned follicles and we stained them for hyaluronic acid. We see that hyaluronic acid increases in this extracellular space, the antrum surrounding the oocyte, and that this increase is statistically significant in the first 6 hours in time with when the follicle expands. We then treated follicles with a drug to block hyaluronic acid secretion. In these follicles, hyaluronic acid is significantly reduced inside the follicle, and follicle expansion is decreased in a dose-dependent manner. And furthermore, ovulation is blocked. So from this, we can conclude that hyaluronic acid secretion drives follicle expansion and is essential for ovulation. Now let's move on and ask what drives follicle contraction between 8 and 12 hours after HCG addition. So we went to some of our early stainings in follicles where we'd seen these actin-rich cells in the outer layer of the follicle when we stained with phalloidin. And we know from the literature that as long back as the 70s and 80s from EM studies, it had been observed that there are smooth muscle-like cells in the very outer layer of the follicle, but whether these were important for follicle development or for ovulation was unknown. So we followed up on this by staining our follicles for smooth muscle actin, and we showed that indeed these actin-rich cells are positive for smooth muscle actin. We then went to our single cell sequencing data and we focused on the smooth muscle cell cluster, where we saw a strong enrichment of genes involved in actomyosin contractility in the period when the follicle is contracting. We focused on the ROCK kinases and myosin light chain 9, and when we treated follicles with drugs to inhibit these proteins, we saw that unlike in the green control traces, where they decrease in volume between 8 and 12 hours, these inhibited follicles actually continue to expand and ovulation is blocked. So from this, we can conclude that follicle contraction is driven by smooth muscle cells in an actomyosin-dependent manner and is essential for ovulation. Now finally, let's look at the final stage of ovulation, rupture, and ask how does the oocyte move out of the follicle? So here is where this detailed confocal imaging came in really useful, where we could basically see how the structure of the follicle wall is changing in the final hour before the egg is released. And we observed that actually follicle rupture can be broken down into 3 steps. So we start pre-rupture, we have an intact follicle wall, and we can see that the follicular fluid marked through this dextrin is maintained inside of the follicle. The first step is then fluid rupture, in which we can see this rupture begin to leak out while the wall is seemingly intact and all cells are still contained. Fluid rupture is followed by cellular rupture, in which the cumulus cells surrounding the oocyte burst out the follicle wall ahead of the oocyte, and we can see through separation with the fluorescent dextrin this hyaluronic acid-rich matrix that encapsulates these cells. And similarly in our cryosection follicles, when we stain for hyaluronic acid, we can always see this mass moving ahead of the oocyte, pushing on the follicle wall. And finally, the egg is released, concluding the process of ovulation. So to present you with a mechanism for mammalian ovulation, we start with a resting antral follicle that's exposed to follicle-stimulating hormone and luteinizing hormone to initiate ovulation. This causes the follicle to first expand, which we showed is driven by hyaluronic acid secretion, and we showed this generates an osmotic gradient between the inside and the outside of the follicle, causing fluid influx and follicle expansion. Expansion is followed by follicle contraction, which is driven by actomyosin contractility in smooth muscle cells in the outer layer of the follicle. And finally, the follicle ruptures and the egg is released. To summarize, we established an experimental system that allows for the visualization of mammalian ovulation at the cellular resolution for the first time. And where we started off with this sketch of how ovulation might look, we can now replace this with an actual visualization of how the process looked in mice. And through this visualization, we identified that ovulation consists of 3 discrete phases of expansion, contraction, and rupture. We identified that these are driven by hyaluronic acid secretion and smooth muscle cells, and could present a model for the spatiotemporal control of ovulation. So this work was done in my postdoctoral lab, so just to acknowledge some people that were involved. Firstly, Tabea Marks, who is the very talented graduate student who I did this work with, Sarah Pania, who did the bioinformatics analysis for the single cell work, and Melina for supporting this work and creating a lab environment where ambitious projects are possible. Finally, in September, I started my lab in Marseille, and just a quick kind of advertisement that it's a beautiful place and we do fantastic science there. So if you're interested in joining, then get in touch. And with that, I thank you for your attention, and I take any questions.
Thank you for the great talk. We have time for questions. Katsurigo?
Beautiful work. So have you tried the change the concentration of gonadotropin, like LH or FSH, in your culture system? Have you changed?
I didn't get it.
Have you tried to change the concentration? How did you actually determine the concentration of LH and FSH?
So one thing that's important to point out is that the real advance we brought with this work was incorporating the imaging aspect into it. But in fact, there were some follicle culture protocols that existed before that we were lucky to have some guidelines to start with. But really, at the start, we were testing multiple concentrations, and we found one that worked well. But certainly, I suppose one limitation of this system is that in the body, these concentrations are transitioning, and it's, you know, we really have a static concentration that starts ovulation, and yeah, this is.
Okay. So if you, I just wondering if you give the excess, the concentration of LH, what's happened if you give the kind of too much LH? Have you tried?
I would say that the concentration we're using is on the high end anyway, so higher than probably it is in vivo. But certainly we haven't done any systematic comparisons of you know, increasing this to something ridiculous. Yeah, and what's also interesting is that we haven't tried taking it away halfway through. So, you know, we don't know whether it's important just to trigger or also to maintain.
Okay, thank you.
Question in the front.
So the movies appeared very violent, and I'm wondering, how does the oocyte experience this? Is it actually cushioned by the cumulus cells, or do you think the oocyte is sensing some of these expansion, contraction, etc.?
So one thing that's important to point out is that we're looking at 12.5 hours in just a few seconds. So of course this makes it look far more aggressive than it actually is. But this we haven't looked into, and it's definitely something interesting, yeah.
The size of the oocyte doesn't change, it doesn't get compressed or anything?
We didn't do comparative measures on this, but for sure it's interesting.
Do we have other questions? Otherwise, I actually do have one. How does it related to the hormones that you also show in the first part? It's kind of like there is kind of an asymmetry between the oocyte, the part of the oocyte which is exposed, or the follicle which is exposed to the hormones, and the one which is more inside the ovary. How does, in your case, does this play out? And the fact that your follicle is isolated in a media which is completely surrounded, I guess, by the hormones, there is no actual concentration-dependent fluids on the different sides. Is this like affects the size where the egg is expelled, or
yeah? So this is a really important point, that obviously you would have a different resistance towards the back of the follicle than the front, and this is one thing that we're kind of looking towards moving back to at the moment. But one thing that's clear is, prior to culture, the direction of ovulation is predefined. So there are a number of kind of markers. So that the wall is thinner in the direction in which it ovulates. We didn't quantify this, but kind of it was fairly reliable that the localization of the smooth muscle cells was more dense towards the back than it was at the point where the follicle would rupture. So this was something that you could always predict, basically. So there's definitely an inherent directionality in the follicle.
Thank you. There is another question in the audience.
Hi, I had a question because I study oocytes in vitro. So I was wondering if in vivo, you know what happens to concentration of hyaluronic acid around the oocyte, if the follicles still maintain high levels until fertilization, if it just lowers?
If the dynamics of hyaluronic acid are the same in the ovary as they are ex vivo, this is the question.
Whether you know or not what happens after the expulsion here. So if the concentration remains high or not of hyaluronic acid.
After the expulsion.
Yes.
So this mass of hyaluronic acid that contains the egg, but also the cumulus cells, is then going to move further down the female reproductive tract, and this hyaluronic acid plays lots of important roles later on. So it's involved in sperm selection, but also aiding fertilization. It's also important for moving through the female reproductive tract. So yeah, it's important that it's not only important for ovulation, basically. It's kind of like a master regulator through various phases of
Right, thanks.
reproduction.
I think we have to move on in the interest of time. Sorry, we can better later. Let's thank Chris again. And our next speaker is Marzia Munafo from EMBL Rome, and we'll talk about precision genetic perturbation during oogenesis.
But I have to wait. Okay. So, making a new organism requires two genomes. However, it's not just two sets of chromosomes that are required, because it is necessary that they come one from the maternal and one from the paternal gamete. And if this is not fulfilled, there is a failure in making the embryo proper or the extraembryonic tissues, which ultimately leads to failure in development. And this is an exquisitely epigenetic phenomenon called imprinting, and really highlights the importance of all the known genetic information that is stored in the oocyte and in the gametes, both gametes, and transmitted to the next generation. So here we're particularly interested in the female gamete, and this epigenetic legacy of the oocyte is established when the oocyte grows in a phase of epigenome programming. But most importantly, it is transmitted to the embryo, where it is present only for a very transient period of time before being reprogrammed. Nonetheless, this known genetic legacy can have long-lasting impacts on development. There are some epigenetic phenomena, such as imprinting, like I just mentioned, or X chromosome inactivation, that are primed during oocyte growth, but then manifest in phenotypes much later in development. So this maternal epigenetic legacy is multifaceted, but in the hacker group, we're particularly interested in one layer of this legacy, which is chromatin modifications. And so the overarching question of my postdoc was really to get to the bottom of the functional role of chromatin modifications in oogenesis and in development. And we wanted to address this long-standing question in an innovative manner, in a way that could be high throughput. So that we wanted a rapid and scalable system, and we wanted it to be precise, because one other question that the hacker group is really keen on addressing is the causal link between chromatin marks and phenotypes and gene expression outputs. So to achieve this precision, we use a resource that we have available at EMBL Rome, which is a collection of mouse models that are catalytic mutants for various epigenetic writers. This means that these mice have single point mutations in the catalytic site of epigenetic writers that kill the catalytic function. And as a result, the protein can be produced, it can assemble into the complex and go on the chromatin, but is unable to deposit the chromatin mark. This means that any phenotype arising in a catalytic mutant can be uniquely assigned to loss of the catalytic function and therefore loss of the chromatin modification. And this is quite distinct from a knockout model in which the protein is removed altogether, and therefore the phenotype is due to loss of the protein, loss of the mark, disassembly of the complex, and so on and so forth. So this is a really powerful system to dissect the functional roles of chromatin marks. But the problem is that in homozygosis, catalytic mutants are embryonic lethal. Therefore, we can't just isolate homozygous oocytes and profile them. So we had to devise a strategy to really conditionally induce this mutation in the oocyte. And to do that, I'm making use of an in vitro culture system that recapitulates oocyte growth, so we can culture growing oocytes and lead them all the way to fully grown oocyte and then metaphase to developmentally competent oocyte. And this is a very epigenetically important phase for the oocyte, because it's the time during which the epigenomic landscape of fully grown oocyte is being established. So if what we want to study is oocyte epigenetics, this is the time window that we want to target. And this system is really powerful because it also faithfully recapitulates what happens in vivo at the transcriptional level. You can see single oocyte RNA sequencing with a beautiful transcriptional trajectory from day 0 through FGOs and to empty oocytes. But what I really want to highlight here is how closely clustered are in vitro cultured oocytes to their in vivo counterparts, implying that this is a faithful recapitulation of the transcriptional dynamics occurring in vivo. And these oocytes are also developmentally competent because when you fertilize them and implant them into foster mothers, they give rise to embryos. And for me, for my specific purpose, the great advantage of this system is that at day 0, we can actually manipulate the oocyte and introduce the epigenetic perturbation of interest. So with the goal of inducing the catalytic mutation in the oocyte, how we decided to do that was to use precision allele-specific CRISPR-Cas9 genome editing. So the way we do this is that starting from a heterozygous state, we microinject Cas9 and an allele-specific gRNA into GOs, and this can specifically program the Cas9 to only target and knock out the wild-type allele and not the catalytic mutant. As a result, we obtain cells that have knocked out the wild-type allele and in which the only functionally active copy is the catalytic mutant, and we call those unmasked catalytic mutants. And the advantage of this would be to have a rapid generation of oocyte mutants, but also the possibility to apply it to any mutation of interest, so not necessarily in the epigenetic field. And as a proof of principle, to validate whether this actually works, I chose ring 1b, which is the catalytic engine of the polycomb repressive complex 1, a conserved epigenetic repressor that deposits H2A K119 ubiquitination of the genome. And at the molecular level, this system, the CRISPR-Cas9 system, is highly precise. All the tested gRNAs were bulletproof, targeting exclusively the wild-type allele. But the really key question was whether it was effective. And as a proxy of whether we are actually making unmasked catalytic mutants, we use levels of the chromatin mark. And the answer was very happily yes. So upon unmasking of the catalytic mutant, we can see a robust reduction of H2A K119 ubiquitination in the nuclei of FGOs, meaning that the strategy is in principle working. And so we really wanted to harness the scalability of the system to actually make a panel of catalytic mutants across a range of 5 different chromatin modifications that are relevant for oogenesis and development. And then as a proxy, once again, we used levels of the chromatin mark to test whether we are obtaining a UCM or not. And in 3 cases out of 4, we could see strong and reproducible loss of the mark, with the only exception being EZH2, in which, despite clear evidence of genome editing, we saw no change in H3K27 trimethylation, which is possibly due to either genetic compensation or a high stability of this mark, which requires cell division to be removed. But nonetheless, this left us with 4 different catalytic mutants, which we can now use to start asking questions about. For example, what happens if you perturb one chromatin modification to the rest of the epigenomic landscape? And so, for instance, we can deplete, in the case of ring 1b, H2A K119 ubiquitination, and look at other chromatin modifications. And in this particular case, we saw that the effect was quite restricted to, of course, K119 ubiquitination, but only K36 trimethylation was affected, which is normally mutually exclusive with ubiquitination on the oocyte genome. On the other hand, when we mutated MLL2 and SETD2, the picture was quite different because we had widespread effects on the levels of other chromatin modifications, implying that setting up the oocyte epigenome requires the concerted action of some of these epigenetic modifiers. But we can also ask what happens to the transcriptome of the oocyte when we perturb one chromatin modification. And to answer that question, I actually generated a single oocyte transcriptomic dataset that spans the 4 catalytic mutants, a range of controls that I won't have time to discuss today, but I'm more than happy to talk about later, and 2 developmental stages, both FGO and M2. And what we really like, one thing we really like about this dataset is that for the first time, we have everything done in the same system by the same experimentalists, in the same sequencing platform, so we can finally compare and contrast mutants, something that wasn't really possible in the past when you would make one flox allele and just profile that particular mutant. And so this comparison enabled us, for instance, to highlight phenotypes for the different catalytic mutants at the FGO stage, M2 stage, or even in embryogenesis. And whilst some of these phenotypes have been reported before, what we're really excited to be able to do now is to really assign them to lack of the catalytic function and therefore to lack of the chromatin mark. And so I will just give you a brief overview, and again, more than happy to discuss later. But for instance, in the case of SETD2, catalytic mutant, we find both at the morphological and at the transcriptional level that there is a defect in meiotic maturation. So the transcriptome of M2 SETD2 catalytic mutant oocytes actually clusters with the FGOs, and amongst the genes that fail to be activated, you have many that are related to cell division and meiosis. On the other hand, for ring 1b, we actually tested what we really care about, which is the inheritance. So what happens if an embryo inherits an aberrant maternal epigenome? And the way we do this is by fertilizing the UCM oocytes with wild-type sperm. And this implies that the resulting embryo is inheriting an aberrant maternal epigenome but receives a completely wild-type copy from the father. And we were able to see that actually this results in an intergenerational effect on 2-cell embryos in which the key gene expression program, which is embryonic genome activation, fails to be activated when the maternal epigenetic landscape is perturbed. So this was a very brief overview, but what I would like to leave you with today is that we've established a new strategy to really generate precise mutations in the oocyte, and we applied it to the study of the functional role of chromatin modifications, and we're able to compare and contrast different mutants to highlight the direct and interconnected roles of chromatin marks in oogenesis and in inheritance. And with that, I would like to conclude by thanking a few people, since this has been quite a large and demanding project. First and foremost, Jamie Hackett for giving me the chance to work in the lab and for being a tremendously supportive mentor throughout the project. Monica, Katrin, and Johanna, who provided experimental and computational support. And most importantly, the facilities at EMBL Rome, the gene editing and the mouse facility, without whom there would be no mouse models and no microinjections. And thanks to the organizers for giving me the chance to present my work here.
Thank you, Marzia. Beautiful talk. Do we have questions from the audience? I will start in the meantime. I was wondering, when you do your Cas9 mutation on the second allele, let's say not the catalytic mutant, this mutation I assume is a full knockout. You're not able to only mutate the catalytic domain.
No. We're not providing any HDR template.
So the question is, once you then fertilize all the oocytes, you will have a mosaic of some carrying the catalytic mutant and some carrying the full knockout. Do you observe phenotypes at 2-cell stage or later which are specifically related to the fact that you are inheriting the catalytic mutant allele versus the full knockout allele?
We haven't followed that specifically. No, we haven't followed that specifically because our interest is the aberrant epigenome from the mother. Then whether they inherit the CM or the knockout allele, it's kind of not necessarily in our interest because anyways they receive a wild-type copy from the father that can kick in as soon as the embryonic genome is starting to be transcribed.
Question, Tom, here in the front.
So the window you're looking is like 10, 14 days, right? Mm-hmm. So from transcriptomics data, you expect to actually have kind of, actually from chromatin modification aspects, you expect to have different modification at different time points, or it's actually, or you expect it to have a sort of switch, so you have a set of modification that then are maintained. You know, because what you are seeing is basically you block a specific modification and then this stays stable. So I was wondering.
It's a really interesting question because there's different dynamics for the different modifications. Some are set quite early, some are set quite late. And so we actually see different phenotypes in terms of hierarchy of each mark affecting the others depending on which mutant we make. And I think that part of it is due to the timing of epigenome establishment, and part of it is due to the crosstalk between the modifications because, for instance, if you set polycomb marks, you might exclude others from those regions, and so it's a crosstalk between two things.
Thanks.
And did you have any of your mutation impacting the growth of your oocytes, or do they all grow normally?
No, I would say in terms of growth, no, we did not observe a phenotype. In the case of G9A, there was a morphological phenotype in the, actually also transcriptional, in the NSN to SN transition, for example.
So I think you showed very nicely that you get a decrease in these marks, but I was wondering whether you also try to look already at specific loci or differences, for example, euchromatin, heterochromatin, whether the mark might be more stable in a certain area, but maybe depleted much faster because there's maybe more turnover in other areas.
That's a very good question. We didn't do it systematically because profiling these marks, I do it with ultra-low input chip and requires quite some material. I have done it specifically for some UCMs that I was following up on and in which I wanted to know what is the effect on the other modification, not on a bulk level, but at locus specific. So.
All right, we'll move on. Let's thank Marzia again for her talk. And now our next speaker is Rose Bulteau from Collège de France, and she will tell us about atomic force microscopy and differences in mechanical properties linked to cortical structure in mouse and human oocytes.
Okay, so hello everyone. My name is Rose Bulteau. I'm a third year PhD in the lab of Marie-Émilie Théré and Marie-Hélène Verlac in Paris and the lab of Clément Campillo in Évry University. I'm working on the mechanical property of mouse and human oocytes using atomic force microscopy, and I'm trying to correlate this measurement with the cortical organization of the cells. So first, I would like to give you a short introduction. So I'm working on oocyte morphogenesis that give rise to a mature oocyte, which when fertilized become an embryos. And this process, the oocyte morphogenesis, is an error-prone process that can lead to miscarriage and developmental disorder, such as trisomy. So here, for example, is the percentage of trisomy in function of the maternal age. As you can see, it increases. In addition, the average age of women at the birth of this first child is getting later and later. And so these elements force more and more couples to turn to assisted reproductive technologies, ART. A common technique used in ART is IVF, in vitro fertilization. However, the success rate is pretty low, only 25%. So that means, in average, couples need 4 trials to get a child, which is a significant psychological burden for them, and it's also an economic cost for the health system. So the goal of my project is to try to find new biomarkers to better assess oocyte quality and try to improve the success rate of this kind of process. So more specifically, in my PhD, I'm trying to understand how oocyte acquires a developmental potential, meaning their ability to become viable embryos after fertilization. More specifically, I'm focusing on the last phase of oocyte morphogenesis, which is the meiotic maturation that lasts 10 hours in mouse, and it's composed of 2 successive meiotic divisions. So during this phase, the spindles start to form in the central region of the oocyte, and then it migrates from the central region to the cortical region, and it leads to an asymmetric division and the production of a mature oocyte at the metaphase 2 stage. It has been shown that a key parameter that affects the developmental potential during this phase is the cortical tension of the cells. So it has been measured with micropipette aspiration technique, where we modelize the cell as a droplet with a surface tension. So the surface tension is the energy required to increase the surface area of the cells. And so it has been shown that the cortical tension decreased during the meiotic maturation and need to be tightly gauged to ensure an optimal developmental potential. In particular, by using engineered oocytes, so for example, the extra soft model, it has been shown that extra low cortical tension is associated with an abnormal number of chromosomes. So all these results show that the cortical tension, and more specifically the mechanical property of the cells, could be used as a promising biomarker to assess the oocyte quality. And so in my PhD, to achieve a complete mechanical characterization of the cells, I'm using atomic force microscopy. So in this technique, a probe interacts with a sample with a specific force, and it creates a deformation that we call an annotation in the cells. And so from this setup, we can obtain force annotation curves, and so we need to analyze these curves to extract the mechanical property of the cells. In my PhD, I'm performing two analyses. The first one is to calculate the area between the approach and the retract curve, so the green area here. And so it represents the energy that is dissipated by the cells during the IFM measurement, so it's linked to the cell viscosity in a way. And the second analysis is to fit a physical model that we developed with Martin Lenz, a theoretician in Paris-Saclay. So this physical model, it's called the elastocapillary model, and it combines the deformation of an elastic component and the deformation of an element under tension that we call the capillary element. So with this expression, we can extract the elasticity of the cells, the cortical tension of the cells, and the capillary annotation rate. Then we apply this methodology on mouse oocytes with non-cortical organization. I mean by that the organization of the myosin and the actin at the cortex. So we work with the different stage of meiotic maturation and also on engineer oocyte, the extra-stiff model and the extra-soft model. So we perform the IFM measurement, and first we obtain the cortical tension of the cells. So the result obtained are similar from what we had with micropipette aspiration, so in a way it validates our approach. And then we also obtain the three other mechanical parameters. I don't have time to detail all of them, so I will directly show you the combination of these four mechanical parameters that we perform through a PCA, principal component analysis. So here on the PCA, so each axis represents the combination of these four mechanical parameters, and each ellipse represents an oocyte population. So here, for example, is the CROR, here is the P1 control, here is the NEBD plus 2 hour, etc. So on this PCA, we were able to observe two regions. Region 1 that corresponds to a stiff oocyte with a thin actin cortex and a lot of myosin 2, so it represents this oocyte population. And in region 2 that group softer oocyte with the thicker actin cortex and few myosin 2, so it represents this oocyte population. So these results show that there is a tight relation between the oocyte mechanics and the cortical organization, and maybe we can use oocyte mechanics to infer the cortical organization of the cells. Then, in my project, we start to wonder whether similar relation exists in other kinds of oocyte population, for example, the case of aged mouse that are known to have lower fertility. So we work with these two populations of mouse oocytes. We measure them through atomic force microscopy, and then I will directly again show you the PCA. So here on the PCA, we keep the previous region, 1 and 2, and we had the young and the old groups. As you can see, for the old group, it's located between region 1 and region 2, and in a way, it could suggest that maybe aged oocytes, they have a thicker actin cortex with different organization than young oocytes. So to test this hypothesis, we performed multiple measurements to characterize the actin cortex, and one of them was to measure the thickness. And we found that indeed, in aged oocyte, the cortical thickness is higher than in young oocyte. So in a way, it showed that the relation between the mechanics and the cortical organization is also present in this kind of population. And more broadly, the maternal age affects a lot the oocyte mechanics. It makes them softer. So then, in my project, the last part was to try to adapt this methodology to measure the mechanical property of human oocytes to see whether similar processes regulate their division as observed in mouse. So we work with the different stage of meiotic maturation, thanks to the collaboration we have with Elsa Labrune, who is biologist in a hospital in Lyon. And so we measure them with atomic force microscopy, and again, I will directly show you the PCA. So on the PCA, we keep the previous region, 1 and 2, with the different mechanics and the different cortical organization. And then we had the 3 stages of meiotic maturation in human. And as you can see, for P1 and M1, it overlaps and it's distant for M2. This overlapping was surprisingly different from what we observe in mouse, where P1 here and M1 are super distinct in different regions. And so in a way, it could suggest that maybe for the 2 first stages of meiotic maturation in human, the cortical organization could be maybe similar. So to test this hypothesis, again, we performed multiple measurements, and one of them was to measure the cortical thickness, and we found that between these two stages, there is no difference in cortical thickness. So in a way, it showed that there is also this link between the mechanics and the cortical organization in human. But the regulatory mechanisms appear to be different, especially for the two first stages of meiotic division in human. So then to conclude, here I show you that we develop an AFM pipeline to measure multiple mechanical parameters in both human and mouse. We show that maternal age affects oocyte mechanics through an alteration of the cortex structure. And finally, we show that human and mouse oocyte mechanical properties are in the same range, with species-specific difference, especially for the two first stages of meiotic division. And then to open a bit the subject, I would like to talk about the microfluidic project that Marie-Émilie talked a bit before. So it's a microfluidic chip with a constriction in the center smaller than the oocyte diameter, so we can really record the oocyte deformation during the whole process. And we show that we can discriminate control control from extra soft oocyte. Can you read the video? Sorry. Thank you. So we can discriminate control from extra soft oocyte without affecting, okay, it's not working at all. Okay, it was just showing that the extra soft oocyte pass quicker than the control oocyte. And so this measurement doesn't affect the oocyte viability. So all this work has been done mainly by Lucie Barbier, a postdoc in our team. And so the next step of this project will be to try to adapt this chip to human oocyte, to try to use in the really long term the mechanic as a biomarker to assess the oocyte quality, maybe in hospital. So then I would like to thank my two teams and all our collaborators. Thank you for the organizing committee to give me the opportunity to present my work. And if you have any question, I will try to do my best answer. Thank you.
We have time for a couple of questions. Yes?
Thank you. So I'm wondering if you measured that. So we saw earlier that there was this poor quality improvement and then poor quality again. Do you see that change in your measurements as well? Like if you look in
the mechanical measurement in function of the quality?
Yeah.
For which kind, like wild type of?
If you look at, well, earlier we looked at human eggs, right? And so they were of poor quality for aneuploid and then got better and then got worse. And I'm wondering if there's any relationship between the two.
So in human oocyte, we didn't check the aneuploidy until now. The human oocyte I'm studying in the IFM study, it's oocyte that coming from IVF, that coming from IRT protocol. So women, they receive hormonal treatment. And then normally with this hormonal treatment, it's triggered the oocyte maturation. And then there is the ovarian function to retrieve the mature oocyte. But in this protocol, sometimes oocyte doesn't respond to the hormonal treatment. And so it's possible to obtain oocyte at an earlier stage, like P1, GV, or like M1. And so it's this kind of oocyte that we are using in this study. So it's oocyte that didn't respond to hormonal treatment, but we didn't check if there are aneuploidy or stuff like that.
Okay, yeah, I guess I was wondering, like, because you showed old eggs and young eggs and what the intermediate would look like in terms of the measurements.
So we know that with age there is more aneuploidy, but in the population that we have here, we didn't check the aneuploidy rate. We just checked the cortical organization, but we can assume that as we know that the aneuploidy is higher in age oocyte, we can say that if they are softer, maybe, they have more aneuploidy,
Questions? All right, otherwise I guess we will move on to our last short talk. Let's thank Rose again. And our last short talk is from Volodymyr Porokh, from Marzia University in Brno, and he will tell us about zygotic spindle orientation.
So, hello everyone. First of all, thank you very much to organizers, to Marie-Hélène and Elvan for organizing this event and giving the opportunity to come to Paris to talk about oocytes. So, we all start our lives as a fertilized egg, which contains two separate parental genomes in a separated structures that we call prenuclei, as Tommaso beautifully introduced. And in this talk, I will talk more about how the precision of this combination is achieved. If you look at the zygotes across different species, you will see that fertilized eggs come in all sorts of shapes, sizes, even colors, and they also come with different developmental potential. If you look at the mouse, it is fairly efficient in reproduction, and most of the zygotes will produce the blastocysts ready for implantation. However, if you look at the human, then you will see that actually in humans, fertilization and the embryo development is more as a game of a Russian roulette, and most of the zygotes will actually fail to develop and produce blastocysts. So this is actually the question that we are mostly interested in, is why some human zygotes succeed and some don't. And in many countries, including Czech Republic, it is not possible to produce human embryos for research. However, human embryos are produced for purposes of infertility treatment in very high numbers, and to sort of use this source of information, we collaborated with the clinical IVF center that also does this time-lapse system during the cultivation of human embryos and records all of the development from the moment of fertilization to blastocyst development. And we got the access to more than 11,000 of human embryo movies when we could actually follow this process in very great detail. However, we sort of wanted to limit the number of movies to analyze, and we focused mostly on cycles where we had the information that this particular embryo resulted in a live birth, so we could say, okay, this is as normal as it can get with in vitro fertilization. And we try to analyze, are there any patterns in human zygotes that will actually hint us, okay, this is a good zygote, it will probably result in a live birth, or is a bad one. Let's not waste our time with that. The first parameter that we wanted to look at is the position of those pronuclei in the zygote, because it was shown in other animals, particularly in mice, that, for example, the site of previous meiotic division actually hints how the pronuclei will be oriented. So we scored the position of our PNs in zygotes, and we actually saw that the position was completely random. We also compared our good behaving zygotes that resulted in live birth with their siblings that actually arrested during the development. This is a CA group, or, for example, failed to implant at the blastocyst stage. And we also saw that actually in those cases, we could not see any difference. So the position of PNs is completely random in human zygotes. However, we did see a pattern, and we saw that if you take a look at the first mitotic division, in most cases it will occur perpendicularly to the axis of PNs. This was true for pretty much all of the categories, but if you take a look at the cleavage arrested group, where embryos only divided a couple of times and then stopped, you'd see that in that group, zygotes tend to have an altered geometry of the first cell division, and they tended to divide in the opposite way as the majority of them. Then when we looked at the second cell stage, we saw that if you look at the nuclear stages of those blastomeres, you would see normally, or usually you would see only one nucleus per cell. However, in some cases, or more so 50% of cases, you would see that one or both of the blastomeres have multiple nuclei, which would hint that something went wrong during the cell division. And we actually saw that the phenotype, where you could see a multinucleation, actually increases with the female age. This is maybe somewhat in contradiction to what was told in the first lecture today, based on the genomics data from trophobacterium biopsies. But we see that in older eggs, you tend to have more multinucleations that would probably hint a missegregation. And we saw that Again, in the cleavage-arrested group where embryos stopped developing, they had a very high incidence of those multinucleations, and also they tended to have those abnormal geometry of cell division. Interestingly enough, if you take a look at the further development in younger oocytes that were less than 30 years of age, you would see that the incidence of multinucleation will decrease with subsequent cell divisions, meaning that the human embryos tend to self-correct those errors. However, if you take a look at the older oocytes that are more than 40 or 45 years old, you would see that the decrease is not really that high, so they don't really correct those errors. So we wanted to also have a somewhat more detailed look at this process, and since we cannot really manipulate with human embryos to do the live imagings, we turned to animal models, and we first started with mice. Again, in mice, we know that this process is highly coordinated, and you don't see really multinucleations. Those are the best behaving embryos ever. And when we looked at the orientation of the first mitotic division, we actually saw the complete opposite of the phenotype that we saw in humans. So most of the zygotes divided in this longitudinal pattern aligning with the PN axis. One of the possible reasons for that is that mouse zygote does not have centrosomes. It has multiple MTOCs that will actually very efficiently catch the chromosomes and assure this precise division. So we went to another model, which is a bovine system, as also Tommaso beautifully introduced earlier. At this point, I would like to shout out to our local ham and sausage producer for the opportunity to get the ovaries from the slaughterhouse. And what we saw is actually that bovine zygotes really resemble human ones. They had a similar incidence of cytokinetic issues during the cell division. They had a similar incidence of multinucleations or micronucleations, although they had a bit longer duration of mitosis, but this is sort of in proportion that they have a longer embryogenesis pre-implantation in vitro. And when we looked at the orientation of the cell division machinery, we actually saw that the phenotype was similar to humans. Again, most of the embryos divided in this nice 90 degrees angle to the PN axis, and we could see the centrosomes inside. And what we wanted to know is if that is actually the same as in humans. So this part of the project was done in collaboration with Professor Mary Herbert in Newcastle, who has a license to actually produce human embryos for research, and we labeled centrosomes in human zygotes, and we could see that 10 hours after ICSI, there is only one dot of SEP152. SEP152 is a protein that actually is incorporated into sperm-derived centrosome and initiates the duplication. So the sperm does not bring this protein. This is a purely maternal factor, and we could see that at 10 hours after ICSI, it starts to incorporate into the sperm-derived centrosome, which is in line with the beginning or on the onset of S phase and DNA replication. And by the 24 hours after ICSI, right before the zygote usually goes into the mitosis, you could see actually two foci of centrosomes in the correct position, close to the interface of PNs. So we narrow down the window when the centrosome duplication actually occurs. Those are the stills from the live imaging. Again, you can see that by the 9 hours, it just starts to become visible, and then around 14 hours, you could see the first centrosome actually traveling across the pond into this final position. We wanted also to know, finally, if the regulation of this centrosome duplication is similar to the somatic cells, where it is linked to S phase, and it is also known that mutations in PLK4 that actually is involved in the centrosome duplication process is linked to some maternal basis of infertility. So we applied PLK4 inhibitor to our zygotes, and in the first movie you can see the control one, where you have a very nice cell division from 1 to 2, from 2 to 4. Whereas when you take a look at the PLK4, then I think you can easily say that the number of centrosomes is not right. We have only one centrosome, and in fact, most of the zygotes under PLK4 inhibition did not really divide, and they did not really also form strong enough astral microtubules to actually push the chromosomes apart very efficiently. So to summarize, we show, and we agree with Tommaso as well, the bovine model system nicely reflects human biology or early steps of human embryogenesis. We show that the sperm-derived centrosome underlies the spatial organization of the first mitosis, and in fact, the centrosome duplication in human zygotes or in bovine zygotes follows the conventional path, and the possible reason for high incidence of mitotic errors in humans might be because of the positioning of an incorporation of sperm-derived centrosome into the cytoskeleton of the egg. Again, thank you for our local manufacturers of meat, and thank you for your attention.
Thank you. Do we have any questions from the audience? Well, I can start with one. I was wondering from the first part of your talk if there is any predictive feature of successful embryo development already at the zygotic level, or all the features starts to pop up after mitosis is initiated?
There have been attempts to find some signs what to score to predict the quality of the embryo. It was way back then in the beginning of the clinical IVF, because in fact they did not really culture human embryos to the blastocyst stage, because culture media were not really efficient or were not really suitable. So people tried to do that, and they tried based on the static observations. So they would take out the embryo day one, they would just look at it and say, okay, it looks like that, and this is a positive sign. But then, since they did not have a really time-lapse system, they could not really see the dynamics. So most of those signs did not really stuck, because it was just basically one observation out of the dynamic process. So the predictive value was not really that high.
Marianne?
Wondering, what do you think is the origin of multinucleation, the tiny nuclei that you get?
This is a very interesting question, because we had quite a discussions with reviewers if multinucleations mean aneuploidy or not. I would say probably not. I would say it's more of a compartmentalizing chromosomes into the nucleus. So I would say that just the machinery that needs to catch all the chromosomes into one single nucleus is not really that mature yet. So you have the normal number of chromosomes is just one, just sort of wanders around and just makes a separate smaller nucleus, and then in the second round of division it will then come together again, and that's fine. This is what we see even in some bovine live imagings. We can see the micronucleus somewhere, and then on the onset of the second mitosis, it just it's pulled inside, and then it's probably fine.
I have two questions actually. What happens to the PLK4 inhibited embryos? Do they make it to the blastocyst with two centrosomes?
No. Like most of them, like 80%, they will not divide. They will try. They will assemble the spindle, because probably the maternal factors are still those that helping, but then they would just like fail into during the cytokinesis. They will just like go apart, but then they will just stay in one cell, and then they will try immediately go into the mitosis and separate into 3 or 4 cells.
So like the video you showed was the kind of a good example because it did the first division. Okay.
Yeah.
And the second one, do you think the connection between the centrosome and the spindle is loose or actually good? You know, like the work from Jan Ellenberg, they kind of see this centrosome often detachment.
We only saw that in a small number of movies. In most cases, it was attached. I would say it's 70% of cases. I had the slide, but I had to cut it. In 70% of cases, the sperm tail still is attached to the pole, and probably is quite important in the next steps as well.
Okay. If we don't have additional questions, let's thank Volodymyr again for the nice talk. And we almost managed to use all the extra time we gained during the coffee break. So we arrive now at the last talk from Melina Schuh from the Max Planck Institute for Multidisciplinary Science in Göttingen. And she will tell us about mammalian oocyte divisions.
Yeah. First of all, I'd really also like to thank Marie-Hélène and Elvan. This is an amazing conference at such an incredible venue, so many exciting talks. So thanks a lot for putting the effort into organizing these events for us as a scientific community. Yeah, my lab is working on mammalian oocytes. And in my presentation today, I would like to tell you about a few examples of how we're studying these fascinating cells, from very fundamental questions, of which many are still unanswered, all the way to application, because we're also hoping that with our research we can help more patients to conceive as they undergo IVF treatments. Now, if everything goes well after fertilization, then roughly 9 months later a baby is going to be born, but unfortunately around 10% of couples are infertile. And this is really a huge fraction. Male infertility, of course, plays a role, but also the egg is very important in the context of female infertility, and in particular age-related female infertility. If eggs are aneuploid, if they carry an incorrect number of chromosomes, then the embryo will inherit these incorrect chromosome numbers. And in the vast majority of cases, the embryo will stop its development really soon. It can't then implant. The woman will then not get pregnant despite undergoing IVF, for instance, or in less common cases, there may be an early miscarriage. And the numbers are really quite shocking. Already in fairly young women, 20% of eggs are aneuploid, and this increases to more than 70% of eggs in women over 40. And for this reason, aneuploidy in eggs is the main cause of miscarriages and female infertility. Now, a key structure that is really important for ensuring that the egg has the correct number of chromosomes is the microtubule spindle. And in mitosis, the spindle is assembled by 2 centrosomes. They are located at the spindle poles, and they help to very rapidly make the spindle within just 30 minutes. In female meiosis, these centrosomes are absent, and the spindles have to assemble by other mechanisms. The spindles are barrel-shaped and quite distinct in morphology from mitotic spindles. So we were very interested in understanding, could these differences in spindle organization contribute to these high rates of aneuploidy, which already in young women are really much higher than mitotic error rates. And Susanna Holubtsova, who now is a PI at Zurich University, as you just heard, she actually worked really hard to establish methods to image human oocyte meiosis. And I will now show you one of her videos that illustrates how the spindle is actually then assembled in humans and how it separates the chromosomes. So here you see an immature human oocyte that still has all of the chromosomes inside the nucleus. Each of the magenta spots here is a pair of homologous chromosomes that has been recombined together into bivalent. And now the oocyte will progress through the first meiotic division and in this way get ready for fertilization. And then first the nucleus breaks down and the chromosomes form a cluster, and then only after several hours the microtubule spindle assembles, and this is a super long process. Over time the chromosomes become aligned, and then eventually the chromosomes are separated, And then hopefully half of them are eliminated into the first polar body, the other half aligns in the second metaphase spindle, and this is now the stage where the egg is ovulated into the fallopian tube and ready for fertilization. But precisely in the step here where the chromosomes are eliminated into the polar bodies, that's where a lot of the errors occur, and there's either too many, too few chromosomes being eliminated with then dramatic consequences for the embryo. Now, what we also observed in the videos that she recorded is that the spindles in human oocytes are very unstable, much more unstable than mouse oocyte spindles, bovine oocyte spindles. And you can see this in this video here, where the spindle is really temporarily multipolar. They do become bipolar at the end again, but even this transient spindle instability correlates very prominently with lagging chromosomes and chromosome segregation errors. And for that reason, we were really very interested in understanding why are these spindles so unstable, and can we do something to stabilize them? And this was done work that Chanceau, Nick did in my lab. He's now a PI at the National Institute for Biological Sciences. And through a lot of work that I can't go through in detail, he screened through mouse oocyte models and combined this with work in bovine oocytes, pig oocytes. We found that human oocytes only have very low levels of a spindle stabilizing protein that is called KIFC1. And this is a kinesin that can cross-link microtubules in the central region of the spindle and thereby stabilize the spindles. And in human oocytes, the levels of this protein are much lower than in mouse and in bovine oocytes. And what we then did is we purified recombinant KIFC1, and what we then found is that this indeed stabilized the spindle, and also the chromosome segregation errors were reduced. And we think that this is an interesting option of stabilizing the spindle that may have positive effects both on the oocyte, but perhaps also on the zygote stages where also spindle assembly is a problem, and even on egg freezing where also spindle instability is a problem. Now we went on to study in more detail how oocyte spindles are formed, and here you can see another unusual structure in the oocyte spindle. This is a mouse oocyte, and in green you see these little droplets at the spindle pole, and these droplets are filled with spindle assembly factors. They are highly enriched in this domain, and this domain is formed by the principle of phase separation. There's a protein that is called TAC3 that is really central to forming this domain. It can phase separate and then recruit a lot of other spindle assembly factors and thereby enrich them in proximity of the spindle, and this is important for spindle assembly in oocytes. Now, I mentioned briefly that there's this unusual phase in human oocytes where the chromosomes are free in the cytoplasm. So the nucleus breaks down, but then it takes some time before the spindle assembles. And this can take several hours in humans. And we were very interested in understanding what is happening in this phase. And here you can see that the chromosomes are moving towards each other into a cluster. And what we then did is we studied this in human oocytes, but also in porcine oocytes, where this also happens because they also don't have MTOCs, a centrosome replacement. And what we could show is that the clustering is essential to prevent chromosome losses and also allow for efficient capture of chromosomes on the spindle. And very interestingly, this chromosome clustering is driven by actin filaments. So in white here, the actin filaments have been labeled, and you see how the actin filaments form throughout the nucleus and then interact with the chromosomes as they come together. And this is a bit reminiscent of what Peter Leonard also found in starfish oocytes. So there's some similarities here, also some differences. One very interesting aspect here in the porcine oocytes is that the kinetochores, which normally associate with microtubules, they can directly also bind to actin filaments as the chromosomes are coming together, and these interactions help to move the smaller chromosomes into the cluster as well. Now, another really interesting function that actin has in the oocyte is shown here, and what you see in green are actually actin filaments, not microtubules. And actually, Marie-Hélène Berlach's lab and also us, we showed that there's enormous amounts of actin in the spindles in mouse oocytes and also actually in human, pig, and sheep oocytes. So this is really widely conserved. And Binyam Mogessi, while he was a postdoc in my lab, he worked on the actin spindle and tried to find out what it is doing. And what he could show is that if you interfere with actin in the oocyte, then the kinetochore fibers are not formed correctly, and then there's defects in chromosome alignment and chromosome segregation. But what we really weren't sure about is whether there might be also a direct role of actin in moving the chromosomes apart. And if you look at this, there's a lot of actin, so that was really an open question then that we were very interested in. And I'm now going to present for the main part of my talk today a project that I'm very excited about, where we investigated this aspect. And it's actually a record project in our lab because it's the project that we've been working on the longest. It started during my PhD time. I then worked on it throughout my group leader time at the LMB, into my now position at the Max Planck Institute in Göttingen. So we've been working on this for a really long time, and only recently we got the tools to figure out what is happening here, and I'll show you this today. So it started with an initial observation that I did in Jan Ellenberg's lab, still as a PhD student at the time. And what you see here are chromosomes in mouse oocytes. This is in meiosis 1. This is bivalent chromosomes. And I was studying non-muscle myosin 2B at the time. And when I stained for it, then I saw that this myosin, surprisingly, was located in the region of the chromosome arms. And you can see here in this merge that it's really more in the arm region. The kinetochore regions are relatively free. And that was very interesting because in mitotic cells, it's not localizing on the chromosomes. And also I could not find it in HeLa cells. So I was really puzzled by this. But it was really tricky to study this in more detail. And one technical challenge was just really that we've been lacking tools. Now, I continued to work on this as a group leader at the LMB, and at the time we looked at how is it localizing in different stages of meiosis, and what you can see here is that in the GV stage, when the nucleus is intact, it's not yet on the chromosomes, but then from metaphase throughout anaphase and into metaphase 2, the myosin is on the chromosome. So then we tried to find out what is this myosin doing, and what I wanted to achieve is to deplete it by RNAi. And at the time we had developed follicle culture for RNAi, so we can culture the oocytes, grow them in vitro, and that made protein depletion much more efficient. But it was still extremely tricky to deplete this protein, and you can see my original Western blots from the time here. So this is the control lane, and here I tried to deplete in follicle culture, but the protein could just not be removed. And indeed, we've now done a study where we looked at the turnover of proteins in the ovary, and we found that there's a lot of extremely long-lived proteins in the ovary. Myosin 2B is one of them, and it has a half-life of 300 days, so no wonder that you can't remove it if you just do follicle culture for a few days. Now, what then really changed the situation, and another aspect was that we could not study the protein in vivo. We did not know which isoform was being expressed in the oocytes, and there it really helped us that we started to work also with bovine and porcine oocytes as a model system. So we found that also in bovine oocytes and in porcine oocytes, the myosin is located on the chromosome surface. So this is now the bovine oocytes, and this is the myosin stained. The chromosomes are stained here in magenta, and the same is true for the porcine oocytes. And what was nice is that we could now also express the GFP-tagged version of non-muscle myosin 2B, and also this localized to the chromosome surface. You see the chromosomes here in magenta, and then in green you see the non-muscle myosin 2B, and the localization indeed looks very similar to the patterns that we see by immunofluorescence. Now, what was then very interesting is that we could really start to investigate how is this myosin actually getting to the chromosomes. And this is the structure of non-muscle myosin 2B. It has a motor domain, it has a coil domain, and also a non-helical tail. And we made truncation constructs and tested which domain is required for chromosomal targeting. And this is what is shown here. So here we express just the motor domain, but you see this is not localizing to the chromosome surface. But when we express the coil domain, you can see it does go to the chromosome surface. The non-helical tail does not. Then motor combined again with coil does localize again. Motor with non-helical tail does not. but then coiled with non-helical tail does. So this shows that you need the coiled region to localize the myosin to the chromosome surface. Now, I mentioned that the myosin is localizing to the chromosomes throughout meiosis, but what was very interesting is that it relocalized to some degree during anaphase and showed a very interesting pattern, which I'm going to show you now. So on the left-hand side, this is the non-muscle myosin to be labeled, and then on the right-hand side, in magenta, that's the chromosomes. Now, when the chromosomes are separated apart, you see that the myosin gets enriched on the chromosome arms, and it specifically gets enriched in the arm region where the arms are separating. And what is interesting is that this myosin really forms like a cap around the chromosomes, as you can see here. And it's not prominently enriched on the rest of the chromosomes, so the kinetochore region is largely devoid of the chromomyosin. And you can see this here as well in these still images from the video. The chromosomes come apart, and you see that it's really sitting in the arm region where the chromosomes are separating away from each other, but the rest of the chromosome is free. Now, myosins normally act together with actin, so we were also very interested in understanding how the actin is localizing. And what you see here is now porcine oocytes where we labeled F-actin. And when I start this video, you will see that also while the chromosomes separate, there's a massive burst of actin nucleation between the chromosome arms as they separate. And that's fairly sudden, and then it decreases again. And you can see this here again in these still images. So in the moment when the chromosomes start to separate, actin nucleation increases, then it drops again, and we see the same also in fixed cells. Now you can look at all three together, at the myosin, at actin, and the chromosomes, and follow how they interact as the chromosomes are separating. And what you can see here now is the caps that are being formed, and then the myosin is really interacting with the actin filaments as they are separating apart. And they all get enriched on lagging chromosomes, as you can see here. So chromosomes that are a bit delayed in their separation, they get enriched in actin and also in the non-muscle myosin 2b. Now, the actin is shown here again, and also actin, you see that when the chromosomes are separating, there is a strong enrichment on the chromatin bridges that is very, very prominent. So this all suggested that maybe this non-muscle myosin 2b does promote chromosome segregation in a direct way. And that's something that we then went on to investigate. Now, the first way by which we approached this is we used an inhibitor, which is called ML7. This is an inhibitor of myosin light chain kinase, and this acts on myosin 2A, myosin 2B. And what you see here in the oocytes when you treat them with ML7, everything is normal, everything looks fine until anaphase onset, which happens at the right time. There's no increase in chromosome misalignment until anaphase onset, so everything is normal. But then when the cells start to progress into anaphase, you start to see major defects. So on the top, this is a control cell. The chromosomes are separating apart fine. But here you see that there is lagging chromosomes, and there's actually also bridges. And we've just been calculating how long does it take from anaphase onset until the chromosomes are fully separated. And this is plotted here. And you see that in comparison to the DMSO-treated controls, the ML7 really increases the time that is required to move the chromosomes apart. And also the fraction of oocytes that have lagging chromosomes are very strongly increased, as are chromatin bridges. Now, myosin light chain kinase also regulates myosin 2A, so we also looked at other myosins to understand are they also on the chromosomes. And this is what is shown here. So this is myosin 2B on the chromosomes, but myosin 2A, other myosins that we've been labeling, we could not find them on the chromosomes. So we think that myosin 2B is the major candidate, but we then wanted to explore this in more detail. And as a first step, we expressed a dominant negative variant of non-muscle myosin 2B, and then analyzed chromosome segregation. And this is now a cell in metaphase. And when the cells are now starting to separate, you see that there's massive chromatin bridges. So the chromosomes really have problems to separate apart. And also here we can quantify this, and we see that there's a strong increase in persistent chromatin bridges, and also the time that is required to separate the chromosomes apart is very strongly increased. Now, everything else is again fine. So we looked, for instance, at anaphase onset, which is normal. We looked at chromosome misalignment before anaphase. This is also fine. And we also quantified various different other parameters of the spindle. So for instance, we looked at how large is the interkinetochore distance. So this is a measure for how strongly the chromosomes are being pulled on. That looks the same between both groups. We also looked at how well are they aligned, how fast are they moving within the spindle. So all of these parameters are the same. So it's really only starting at anaphase onset that we see these problems. Now, how is it in bovine oocytes? So also here we see both the fixed cell, live cell myosin on the chromosomes. We express also the dominant negative variant. And also here we see an increase in lagging chromosomes, and this is actually work that Tommaso did still in our lab. And we see an increase in lagging chromosomes and oocytes with chromatin bridges. So also this is very prominent in bovine oocytes. Now, coming back to the mouse situation, what about mouse oocytes? And to investigate this, it took us a really long time. We established a knockout approach where we depleted the myosin because it's so stable in the mouse, where we depleted the myosin in a really early stage of oocyte development with a SPO11 tree. And here in the top, you can see a control spindle. Everything is fine. And then when we look at the knockout oocytes, we also did see an increase in chromatin bridges in the oocytes as they progressed to anaphase, although also the other parameters of spindle assembly, anaphase onset, they were normal again. So also in the mouse, we see these defects. Now, what is very interesting is that ML7 is not causing chromatin bridges in mouse oocytes, and that means that the myosin to be is likely regulated by a different pathway in mouse oocytes as a model system. So to summarize what I showed you, we found that in mammalian oocytes there is an enrichment of non-muscle myosin 2B on the chromosome arms. It's particularly enriched in the arm region where the arms are associating. And then it gets even more enriched as the cells progress into anaphase. Also, actin gets enriched in between the separating chromosomes. When we interfere with non-muscle myosin 2B function, the chromosomes struggle to separate away, and that's why we think that this myosin 2B, which you could refer to as a chromomyosin in analogy to chromokinesins, helps to separate chromosomes apart. Now, why do you need a myosin to separate chromosomes in meiosis? Why don't you need it in mitosis? So we can only speculate why this is the case. But an interesting difference is that in mitosis, there's only a very small interface that needs to be separated. You only have cohesion in the pericentromeric region, and you need to separate the chromosomes in this region. But in meiosis 1, you've got this really large interface where meiotic recombination has occurred and where a lot of cohesion, a lot of DNA needs to be separated apart. And it's very possible that this difference in geometry may make additional mechanisms necessary that help to move the chromosomes away from each other. Okay, so finally, I would like to thank the people in my lab who've done this work. Susanna did the human oocyte live imaging. Chanso did the work on spindle stability. Binyam on the actin spindle. Ina was driving the bulk work of the chromomyosin project. And yeah, I'd like to thank you for your attention.
All right, questions?
Hi, thank you for the talk. Two quick questions. Have you tried to trim away myosin 2B?
Yeah, so we have, that's actually something that we're doing at the moment. So it's something that we're testing in the bovine system, but we haven't yeah, that's ongoing.
And my second question was like, have you tried to look at myosin 2B localization, but like in a cyto-D complex when you like depolymerize actin, and you don't have like spindle actin, for instance?
Sorry, if it's localizing, yes, it's independent of actin functionality, okay. The localization, yeah.
Thanks.
Really cool. Thank you for sharing it. I also have two questions. One was, have you looked at its role in meiosis 2?
Yeah. All of these experiments, as you probably know, are extremely time-consuming and difficult. So we have not done activations in meiosis 2. In meiosis 2, although I think so, our model is it helps in meiosis 1.
Yeah.
I still think it may also be required in meiosis 2, just because you have to install the system in the first instance, and it's still there, so it's kind of part of the system. So I wouldn't be surprised if it's also required for meiosis 2 activation. We haven't tested this. But yeah, it's something interesting to look at.
And then my other thought was that it reminds me of inhibiting TOP2 with these chromosome bridges. And so are you thinking that it's somehow helping with like entanglements?
I definitely think so. So I could imagine that you've got like maybe topoisomerase, can only do so much, and you have to maybe really have additional kind of force of separating everything at the same time. And how precisely it interacts with other pathways, we don't know, but I can easily see that the geometric situation is really quite different in meiosis compared to mitosis.
I found the localization really interesting, that it localizes to the ends somewhat of the chromosomes, in particular then to chromosome bridges as well.
Yeah.
So can you speculate what it might recognize there? Because clearly it's not just chromatin, it must be something else on the chromatin on these areas.
Yeah. That's a really good question, and we don't know. It could be that it's actively excluded from other regions and then just binding there because maybe the surface properties are different, but we don't know. It's difficult to know what could be good candidates.
So what you showed remind me a bit of what happens in nematode in meiosis 1 in Julien Dumont's lab, where he showed that you have this pushing of the chromosome by microtubules that are nucleated. Do you think that actin there with myosin are actually pushing this chromosome apart when they have difficulties to segregate?
I don't know. I mean, I would think that because there you can screen so well genetically that it would have popped up probably, but I don't know if there might be other mechanisms that then allow for more efficient degradation or maybe some kind of more clearing of cohesins or something like that. I don't know, but it could be that it's undiscovered, but at the same time you can screen so efficiently that maybe it would have popped up.
Additional questions? Yes?
So I like the idea that in meiosis you have sort of an additional problem to solve, but that would be true in male meiosis as well. So in your SPO11, that would affect male and female.
Sorry, in the what?
You had a SPO11 conditional knockout, right?
Yeah.
That would affect both germlines. Can you say anything about male meiosis?
That's a very interesting question. We have not tried to breed the homozygous males, but that's a very interesting question, actually. Yeah.
Or maybe there's something about the oocyte that makes it especially needing that. I don't know.
Of course, there's differences in recombination patterns and so on, but I think that's definitely interesting. I think to study the chromosome segregation spermatocytes, it's a little more challenging, but I definitely think it's interesting.
Not with all the fancy tools you have of like live imaging, but just to look at them. Did you see bridges and all this stuff?
Yeah. No, I think that's a good point.
Just to follow up on that, because I think if the size of the oocyte could be a factor, and then also that you have to, because actin ends up in the polar bodies too, right, in some species at least, so if they're kind of directing all of that as well.
Yeah, I mean, so maybe whether there's also function in somehow making sure how the chromosomes are getting pushed into the polar body. Yeah, I mean, I think it's definitely also a huge spindle, right? So it's a fairly large distance that they have to bridge and move apart and do it quite rapidly. So that's definitely possible that also these, you have to separate really quickly, make sure that the cytokinetic furrows ingress after separation has been completed. That's definitely a possibility also. Mm-hmm.
Sorry, maybe it's a silly question, but have you tried to get rid of microtubules at this stage?
To do what?
To get rid of all the microtubules at this stage.
Yeah, I mean, the actin spindle, nothing happens without the microtubules. So you still have to have those in the backbone and the actin spindle. As soon as you take microtubules out, the actin filaments are gone as well, and then you build it up again, and then they are just there again. So it's still a system that relies on microtubules in the background.
Do you have any last thoughts or questions? If not, let's thank Melina again. And I'm going to pass the mic to Maria-Helene for some final remarks. Thank you for being here this afternoon.
Could I have my thanks slide so that I don't forget anyone? Okay. So, well, I really wanted to thank all the speakers, all the participants. I think we had wonderful talks, beautiful questions, and I really enjoyed the meeting. So thanks to all. I want, of course, to thank the Fondation Saint-Ger-Polignac, and in particular, Professor Pierre Corvold, who is actually at the origin of this meeting happening, because he asked me to organize one, because he's the president of the Fondation Saint-Ger-Polignac. So thanks, Pierre. And thanks, Céline Campa from the Fondation, who also allowed this meeting to take place. Thanks Maya Regui from the CIRB at the Collège de France, who took care of hotel booking and restaurant for the speakers. And I would like to thank all our sponsors, so the Company of Biologists, the EMBO YIP, who actually agreed to pay for Yaniv El-Koubi's trip, but unfortunately they couldn't come. The PSL Q-Life, the MemoLife LabEx, and the Société de Biologie Cellulaire de France. So all of them contributed to finance the hotel and the restaurants. So, voilà, thanks to all of you. And yes, just a little reminder for the speakers. So the restaurant is booked at 7:30, but you can arrive at 7 if you want to have a drink before. No problem. And you have the address, I think. It's Restaurant Le Bon.